Imagine that you and your family are living a perfectly happy life. When all three of your children are spontaneously faced with a fatal, callousing heart condition, you feel like a terrible parent for letting this happen, but there's absolutely nothing that you can do to make it better. If you think that this devastating situation sounds like something straight out of a Grey's Anatomy episode, then you're correct.
Yes. Genetic profiling is a rapidly growing science. New findings crop up every single day. For example, 3 months ago, there were only 33 known genetic causes for cardiomyopathy. Today, there are 34. This right here, this is the genetic mutation that gave your kids cardiomyopathy.
It isn't the water, it isn't anything.
Thanks to the ever-evolving research in genetics, Sabine was finally able to find out what had harmed her family like this. The question is, how many other people can be helped? And to what extent are researchers actually effective in determining a person's inclination to having or developing a genetic mutation? To answer this, we first need to understand the ways in which we can read genetics. What we call genetic sequencing is the process of reading the bases in our DNA, going from 5' to 3', and consolidating the code into a full or partial genome sequence. On the other hand, genotyping uses a previously sequenced model of the same geographical area of the genome as reference to compare the outlier base pairs to, which are known as single nucleotide polymorphisms, or SNBs. Because the majority of our genome carries information for basic cellular functions needed to keep us alive, the SNBs are the nucleotides that visibly differentiate us from one another in terms of ethnicity, build, and medical conditions. When comparing these two methods, we need to keep in mind that the human genome is 3 billion base pairs long. Of that, only about 3% are genes, coding for proteins, which we have about 20,000 of, each one averaging out to be about 3,000 nucleotides long. You can only imagine what a tedious job sequencing all of this would be, which is exactly what the Human Genome Project finished in the early 2000s. And the entire job took about 20 years, considering that it was an international effort with 20 countries involved. We needed a shorter way to do this process if we wanted to keep advancing in this field. We also needed to preserve our sanity, because the most common way to read DNA was with the Sanger electrophoresis method. In this procedure, the DNA is split into sections with the endonuclease enzymes that work the magic of hydrolysis to cut backbone, and then it's amplified in a prokaryote. After isolating the plasmid DNA, we place it into a solution of primers, free-floating bases, some fluorescent terminator bases, and replication enzymes like polymerase, to then be heated and cooled multiple times, aiding the enzymes and DNA to denature and replicate over and over again. The enzymes will start at the Tata block and add bases to the leading strand, eventually ending the segment with a terminator base. After the solution is given an electrical charge, the sections of DNA will flow through a polyacrylamide porous gel in a capillary tube, arranging themselves from shortest to longest along the way. A camera will detect the fluorescent tags, and the results will line up with one another to agree on a common code for that DNA segment. In short, multiple trials of the same process have to be completed in order to have an accurate result, which is absolutely exhausting when it comes to a large amount of code that needs to be read. We can now see why industrial genetic testing companies choose to genotype by SNPs, or exomes, instead of doing a full genome sequence for their customers. What's neat is that the single nucleotide polymorphisms give us that very exact information that we're looking for by comparing our base pair outliers to the ones of our very, very old ancestors from various continents and descendancies. That must have sounded really confusing, but basically speaking, genetic sequencing takes a whole lot of effort and time and resources, which is why in the past couple of years, we've made it easier to do. And many people now rely on the invention of fast, modernized genetic testing to determine their ancestry for recreational or, more importantly, medical purposes. We consider this information to accurately represent us as people, but to what extent is it actually correct? Well, for starters, the curious recreational tester should know that there is good reason that speaking in terms of chance is the most accurate way to go. while companies like Ancestry and 23andMe use common ancestry informative markers from each major race of people, the genetics databases for smaller regions and communities in the world vary significantly. In efforts to keep the businesses running, they refuse to agree on the common database, making their customers question the test's authenticity when they compare both results to one another. As you will notice when your genetic test results come in, there's a section dedicated to your personal health, diseases, and mutations. These are detectable because of the single nucleotide polymorphisms yet again. The problem is, no matter how many different techniques are applied to finding the SNBs, they are still incredibly hard to identify and take note of because they appear once every 200 to 300 bases. The ones that are found may not be the only ones, and their concentrations differ a lot from one chromosome to the next. These many factors make us question our decisions when the choices we make are based off of this cheaper and faster type of evidence and testing, not always being suitable to base our medical trust into. Of course, this is revolutionary technology that will change medicine completely, but having the cardiomyopathy gene does not mean that you will die, and not having it does not mean that you're inherently safe either. In the end, the accuracy of genetic testing is highly variable. For example, the Human Genome Project admitted their error rate in the year 2000 to be 1 in every 1,000 base pairs, later improving it to being 1 in every 10,000. This level of accuracy would only be achieved after retesting the same segment 3 entire times. Even the cell itself makes mistakes every billion nucleotides, but luckily the polymerase enzyme, the DNA builder, also acts as a proofreader for your genetic strands. To summarize this video, we have talked about the difference between genotyping and genetic sequencing, the most standard way to read genetic code, the uses and demand for transcribed genetic information, and how accurately the sequencing business meets the standards of the customers, scientists, and our personal health. A deeper understanding of how this type of science works gives us the opportunity to ponder the process, even though predicting our accuracy with it will always be subjective and very open-ended. Nonetheless, I hoped you learned something from this video. Thank you for watching.